So let's switch to a different topic. So I'm a physiologist, and I looked at stress marker in fish. And this is actually my PhD title, which I submitted yesterday. And yeah, I hope you enjoy. And so let's start. So what I've actually done in my PhD is like looking at different stressors. So the human impact on rivers is like very diverse, and it's very like multifaceted, and we have a lot of impact from things we don't consider. And what I've done is looking at how the fish actually reacts. So when there is like there's a specific tolerance till the fish doesn't react, and at one point there's a reaction, and we will see like a response in the fish. And the main marker I looked at in my PhD was cortisol, which is released by the hypothalamus pituitary internal cell axis, and oxidative stress. So oxidative stress is an imbalance inside the cells, and we have the main important things we have to consider now here is like we have antioxidants which trying to balance the free oxygen radicals, which I looked at, and we can have, if there's not an if there's an imbalance, we can have the stress of the cells can destroy lipids or proteins, or we can even have DNA damage. I would like to start to divide my PhD in two chapters to make it a little bit more easier to follow. And the most obvious pollution we have is chemical pollution. So the first chapter will deal about pollution. And the second one is actually light pollution. This is the River Rhine. This is in Düsseldorf. And you can see actually how much light is coming from all the city and influencing the river. But first of all, let's start with chemical pollution. And during the first year of my PhD, I was doing a biomonitoring on PFAS pollution. So PFAS is a perfluorinated aqueous substances, and they are released from a fluorochemical plant in the river system in Veneto, in the region in Italy, where I'm actually based. And the pollution is one of the biggest pollution in the world. And we went to 3 different sites and just sampled 2 different species, one Bentivorous and one Potodromus fish species. And actually the 3 sites we selected, this is, you can see 582.6 nanograms per liter of P4I. It's not the highest polluted site. So we have even sites which go up to 1,200. And what we have seen, so first of all, we analyzed the PFAS concentration inside the fish. And due to that you need a lot of tissue for doing this kind of analysis, 3 grams, and if you do biomonitoring studies, of course, you want to have the impact as low as possible. So we pooled the Patagobius, the small fish species, and we analyzed the whole body concentration. But in the other species, in Squaliocephalus, in chub, we could actually do the division. And we see that there is a much higher concentration in the liver. And why? Because liver is very protein rich, and it's also the first instance of defending against chemical substances, so it makes sense. But we can also see like the concentration, they are going up to 300 microgram per kilogram of fish weight, and this is actually, I read a lot of literature, this is like concentrations I've never seen in fish anywhere in literature, and this is very concerning since we didn't go actually to the highest polluted site. So but I'm not really interested in this. This is a biomarker of exposure. I am interested in biomarker of stress. So what we actually did is like analyzing the cortisol concentration in different tissues. We analyzed cortisol in the serum of the fish, and in the muscle, in the fin, and in the scales. And you can see, so the dark is the Squaliocephalus, and the other one, the light bars, they are the Patagobius ponelli. And we can see that the Squaliocephalus is following through all the tissues the same pattern. So there is a highest statistical significant increase of cortisol production in all different tissues we measured when there is PFAS. But we don't see this in the other fish species. So it means probably Patagobius ponelli is not stressed. And this is actually a big problem because if we have organisms which react to kind of chemical pollution and other organisms which do not react, it might disturb our ecological balance. But it didn't seem that the fish didn't react to the PFAS pollution, because if you look now, this is like liver histology, so I made some small cuts of the liver, and then you stain it, and then you can see there's like white spaces, and this is actually when there's more fat. So this is liver, like the vacuolization of the cells, and there's more fat inside the cells. And we can see this, the first fish is Patagobius burnelli, and actually they increased the amount of fat inside the cells when there is the PFAS pollution, but we didn't see any reaction of the other fish species. So now, coming to the ecological implications of this, might it be, this is a question, might it be that Patagobius actually is adapting to this PFAS concentration? This would be very bad, because we have, as I said before, if there's an imbalance of species, we will like change the whole ecosystem. So this is definitely something we should further invest. There's much more interesting research and results. We also published some of the results already. The rest, the cortisol results, will be published soon. They're already under revision. Yeah. So, but now we will focus on the second problem, the light pollution. And we did a study together with Velisara, and we had 1 and 6 fish, and we exposed them to like a light bridge setup. So the fish, they were like simulated for 90 minutes. And then we had in these 90 minutes like 2 times where we like switched on some light simultaneously, like when fish were like swimming in a river and they were like crossing 2 bridges. And I divided this setup into different stimulus to like compare, like, because we implemented the fish some heartbeat tracker, and to actually capture how the fish react to this light pollution, I divided the 90 minutes into pre-stimulus, then the actually stimulus time, where we had like 2 times the light flashes, and like post-stimulus. And what we actually can see in the heart rate is, so let's start with the first fish, is that when there is this light event, which we can see here, I don't know if you see my pointer, no, I'm so sorry. So when you see there's like this sun, this is like the light, and we can see that there is, when there's single fish, the heart rate variation. So this is the heart rate variation, which was calculated by the resting heart rate. So the fish, we did the surgery, and they were like recovering from the surgery, and then we had like 5 days before we started the experiments actually, and this heart rate I used like as a standard heart rate to actually normalize my model, which I did here. And we can see that in single fish, they highly react. So there's a significant difference when there's like the stimulus, and then we see after before the stimulus, the variation is higher, and afterwards it drops. But we don't see this in the 6 fish. We can assume there is something, but we don't see that there is a reaction actually. And this actually aligns what I've seen in the cortisol response. So when fish are alone and they are exposed to the light stimulus, they react. So there is a reaction. But this is measured in the muscle. We also measured cortisol in the plasma of the fish, and we don't see this in the muscle, in the plasma. So what does it mean? Actually, what I assume, so this is measuring cortisol in the muscle is quite new, also because normally it's just measured in the plasma. And what I assume is that actually the cortisol response is delayed. So when we see the cortisol in the plasma, it's much more recent what actually happened. So there is a reaction to the light stimulus actually, but we don't see it in the plasma. But what we see here now is also very interesting. 6 fish without light, they have the highest stress levels. And I was like, why? But if we continue looking at some other data I obtained, so this is on the left, the heart rate variation. So how much the heart rate varies through all the experiment. And heart rate variation is also an indicator for how fish can react to actually a stressor. When the heart rate variation is low, it means like there's a low reaction of the fish to react to a stressor or to an external stimulus. And the second graph we see, this I selected because I like a lot, it's easy to see. Here we have the 6 fish, And there's actually, this is different antioxidant enzymes, where I looked at the gene expression, and we can see there is a higher oxidative stress potential in the 6 fish without light treatment. And then, why? So let's come to the reason why we see this. So here we see the 6 fish, and they do something, they are trouts, they do something very normal. They like to build up hierarchies. And actually, when they don't have light, they are much more tend to build up these hierarchies. And this is also confirmed if you look at adenylate kinase activity. Adenylate kinase is for us like now how much we have heart activity actually inside the heart. And we can see that the 6 fish, they have much more energy spent in the heart. So it confirms actually the hypothesis that 6 fish, there might be a light stimulus, but the social stress is much bigger for them than the light stimulus itself. So this is the first chapter. Now we have to continue with the second chapter of my PhD. So now we will speak a little bit about downstream and upstream migration, how we actually can invent my physiological measurements into this. And the first thing I would like to mention is like the experiments I had with Gloria, like we exposed. I don't think I have to explain the experiments again, but this is the results I obtained. And we had 1, 2, and 6 fish. And this is a nice video for my perspectives, because we can see that there's this single fish, like impinged at the downstream grid. And as soon as the group gets closer, he gets kind of like motivated. So he's like joining the group after some seconds, as you can see. And it's very interesting because it aligns with my results. So when they are alone, they are much more stressed. So this is the protein oxidation, and this is the oxidation of lipids we can see. And we see that single fish, they are stressed. But even if they are by 2, the stress level is much lower. And we see the same pattern also in the cortisol response measured in the muscle. And what does it mean? So in an ecological context, it means, okay, fish can be alone, but if we have to consider designing fish passages, it's better even if they are by 2. And so they have a better passage. And especially this study has a very important ecological implementation in the sense of this is an endemic species which is not known to swim that much, but for like designing fish passages, for designing for making actually nature conservation and for protecting actually animals, it's also very important to consider species which are not known for their migratory behavior. And why? Because otherwise we get like inbreeding, and we get like much more disease spreading, and we have the problem that like species will like distinct just because they cannot move. And now we are coming to the last experiment I performed. I was together with Velisara, and we were in Vattenfall, and we exposed salmon smolts to like a jet, to an additional jet, with the idea that fish will guide around hydropower plants. And we had like 3 treatments: a control treatment, a right treatment, and a left treatment. And what we actually seen is that the oxidative stress is much higher when fish are treated with the right treatment. Why? I mean, the jet should be like, the flow of the jet was same. And we see the same. So this is oxidative damage. We see also that there's a response in the antioxidant defense system. So there is an increase of GPX and catalase. So we actually confirmed there is much more oxidative stress. There is not only more oxidative stress damage, which we can see in the lipids. And now we have to ask why we see this kind of analysis in the lipids, because we didn't see any difference in the proteins. I work with red blood cells. So red blood cells, they are known to have like a very protective membrane, which is very thick in fish, and they have a lot of lipids inside this membrane. And so actually there is more energy consumption, there is more oxygen which is actually needed from the fish, and we have this moment where the lipids are destroyed. And why not proteins? Because proteins are much more protected from the cells. So if you see something in the proteins, it's much more urgent than in the lipids. But we see actually, as we can see here, there is antioxidant defense. So why? Now is the question, why? So the jet came from right, and one possible explanation is actually, so the fish swims and it's pressed in the inner channel and they don't like. This could be a reason, but the other reason could also be maybe there's like a stimulation on the lateral line, so actually maybe the angle of the jet also matters. So because we don't have actually the distribution how the water was coming inside the flume, and maybe it's like only small changes already can like impact the fish. We should definitely invest more in this kind of research, because it seems to be very interesting. So now coming to the discussion and to summarize a little bit all my all experience during the PhD. So first of all, I really appreciate working in a team, because it's like you get like specific angles, especially working with engineers as well. Like you don't consider all the ideas, because we are sometimes, I work a lot in the laboratory, so sometimes you don't have like this, you lose a little bit how to work applied, let's say. And the second thing is sometimes marker, like if you want to look at a stressor and you want to see a reaction, it's better like consider like many things and then see, because sometimes as I've shown in my results, cortisol, for example, is known as a major stress hormone in fish, but sometimes cortisol didn't show any changes, but oxidative stress did some, or even the other way around. So multi-biomarker approaches are very nice. And also, this is something I didn't mention, I really like working in these semi-controlled environments. So working with wild fish, but like trying to have all the other parameters controlled. Because if you do biomonitoring, it's nice and it's interesting, but it's very difficult to publish because there's many parameters which can actually, even though we have the highest concentration in PFAS, there's many parameters which can actually influence our results. And the fourth one is, of course, communication. Communication is very important, especially working in a team. And the last one, this is like the conclusion of my PhD, which I just added. My PhD was like called biomarker, and Gianfranco and me, we had a lot of discussions, because biomarker is nice, but I don't think that there is a specific biomarker which can describe the feeling or the welfare of the fish. It's more like you have to adapt the marker you want to look, or the things you want to look to a specific physiological question. Yeah, and also especially because responses are very species specific. My colleague, she's working with Antarctic fish, for example. She sees completely different responses, even though they are fishes in respect to antioxidant enzymes. So, and now I would like to thank everyone, and also especially all the ESRs, and this has been a very great time with all of you.